and my name is Vanshika. We are the podium coordinators for this deep thought. Before we start, we'd like to request everyone to put their phones on mute.
We'd like to introduce the judges for this round, Dr. Raghav Rajan and Dr. Anuradha Ratnaparkhi. Dr. Anuradha is a senior scientist at Agarkar Research Institute, Pune. She studies cellular signaling mechanisms in Drosophila melanogaster, focusing on synaptic signaling and glial morphogenesis.
Dr. Rajan is a professor at IISER Pune. He studies the neural events that lead to the song of a songbird. We thank the judges for being here with us. We request our judges to pick a chit and determine the first team to answer the question. We will proceed clockwise from them. So we'll be proceeding with IIT Delhi, then IISc, followed by IIT Madras and IISER Bhopal.
We'll now begin with the biology deep thought. Presented on the screen are the 4 questions for this round. IIT Delhi, please choose a question.
Too many A's on my transcript.
Too many A's on my transcript. Santa Sharanya is studying the genetic regulation of the synthesis of the newly discovered amino acid, IISER, in the bacterium Diptybacter. IISER is found to be a very relatively rare molecule in the environments of the bacterium and is energetically expensive for it to synthesize. Observation 1. Through some preliminary screens, Santa Sharanya observes that the synthesis of IISER is inhibited by high concentrations of IISER in the cell. That is, IISER inhibits its own synthesis. He wants to find out the mechanisms that help the cell achieve this. The genetic machinery involved in this regulation is isolated and is as follows. IISER PSH, promoter and operator where RNA polymerase binds and starts transcribing. IISER BNR, the genes that synthesize the machinery to make IISER. IISER MB, the region between PSH and BNR. Transcription and translation are simultaneous and happen at the same rate here. That is, ribosomes start translating a mRNA transcript while it's being made, and translation of each gene happens independently. Note, the rate of translation is dependent on the intracellular abundance of the respective amino acids, and there is only a finite availability of ribosomes. Observation 2. Santa Sharanya finds that IISER can form a complex with another protein, blue molecule in the diagram, and this complex can bind to the IISER PSH region and reduce the ability of RNA polymerase to transcribe. However, even when this binding is chemically blocked in IISER-rich conditions, IISER synthesis is found to be negatively regulated. Observation 3. In IISER-rich conditions, Santa Sharanya observed many short mRNA fragments of 140 bases, whereas in IISER-deficient conditions, the fragments were of 7,000 bases in size. This leads Santa Sharanya to suspect that something is happening at the transcriptional level. He looks at the IISER MB region. The complete mRNA transcript of IISER MB contains regions marked 1, 2, 3, and 4 that can bind to each other and form loop-like structures. The region between 1 and 2 contains a stop codon at which the ribosome stops and falls off. During transcription from DNA to mRNA, a loop followed by a string of A's destabilizes the DNA mRNA structure and separates them, effectively terminating transcription. These are the 2 possible structures mRNA can be in. Observation 4. Through biophysical measurements, Santa Sharanya finds that mRNA transcript exists in 2 structures. 1. Region 1 binds with region 2, region 3 binds with region 4. Or 2. Region 2 binds with region 3. Observation 5. Santa Sharanya also finds that the codon sequence of IISER MB has 2 successive IISER codons in region 1, which he finds odd given the rarity of IISER. Question 1. Given all of the observations, propose a model for how the IISER MB element is involved in regulating the synthesis of IISER. How does your model explain observations 2, 3, and 4? Question 2. What is the significance of the 2 successive IISER codons? What could you infer if in a similar regulatory system for another amino acid, there were 7 such successive codons? Santa Sharanya finds that the first mode of inhibition, IISER binding to IISER PSH, of isosynthesis is dominant at mild to moderate levels of ISO deprivation, whereas the second mode, transcriptional regulation, is more dominant at more extreme levels of deprivation. Question 3. Given this observation, propose an explanation for why Diptybacter requires two regulatory mechanisms for isosynthesis. That is the end of the question. Participants, if you have any doubts, you can ask them now.
Can you clarify on the statement that isosynthesis is found to be negatively regulated?
Oh, yeah. Yeah. Should I repeat that? Yeah. So if there's a lot of ISER in the cell, you would not want to produce any more machinery to synthesize ISER. So that is what I mean by negative regulation.
If there are no more doubts, teams, your 15 minutes of thinking start now. Participants, you have 10 minutes remaining. You have 5 minutes remaining. Participants, you have 1 minute remaining. Thirty seconds remaining. Participants, your time is over. IIT Delhi, you have 5 minutes to present your answer. Your time starts now.
So in the for observation 2, we can a simple explanation could be that laser that is present that simply uh, negatively affects the its own synthesis. So the middle portion actually actually just stops synthesis of the amino acid. Uh, for observation 3, we can see that the observation for 3 and observation 4 are quite related, as we can see in the from the mRNA, the two regions, the two transcripts that can be formed are one of them is not so stable and just breaks into the and just breaks because of the AAA sequence from the portion, and hence that's probably the reason we can say that the of out of in general 7,000 bases base sized mRNA just break down into the 140 bases. As far the second question is concerned, the two successive ISR codons are probably required to synthesize more mRNA, and henceforward the more amino acids through translation. If there were 7 such consecutive codons, I believe it will be more regulated, and hence more such amino acids could be produced.
So first of all, for the third question, I think the reason that we need two regulatory mechanisms for ISR synthesis is given that how expensive it is to synthesize ISR. um, if we had the same mechanism which works at very extreme levels of like deprivation, also working at the mild to moderate level, then it might lead to a positive feedback loop, or might lead to excess production of ISR than what we need. Therefore, having one that works at extreme level and then stops when you reach mild to moderate, whereas another which serves your needs a different range is a better bet. Secondly, some preliminary ideas on observation 3 are that if you have an excess of ISR, right, when the mRNA is being transcribed, then we see that we have shorter mRNA fragments. So my hunch is that the tRNA, when it reaches the two ISR codons, it does attach the like tRNA on top of it, because of which one becomes unsuitable of forming loops. As a result, we get structure number 2, which you can see over here, does have a stop codon. Now, since we know that this entire thing, we are assuming has approximately like 7,000 base pairs, which matches our observation as prior, we would expect that this is the structure which is present in ISR deficient conditions, which makes sense given that here we have the two codons and there is nothing bound to them, so the ribosome cannot go past them, and they're free to bind and form this structure. Here they are not free to bind and form the loop between 1 and 2, which is why the 2 and 3 loop forms and it gets cut off over here, and this is the 140 base pair thing which is observed. This or this along with the PSH observation.
You have 1 minute left to present your answer.
So like again, by this process, we can see that this is clearly negative feedback loop, which explains observation 2, why even in the absence of the chemical thing, this is still being inhibited. Observation 3 is also explained by this. As for observation 4,
30 seconds.
yeah, so the explanation of that is the fact that whether or not tRNA are attached, like if tRNA are not attached, this is stable, otherwise tRNA attachment is more stable. So the amino acid brought by the tRNA, that is the more stable bond than hydrogen bonding or whatever is holding this together. 10 such successive codons. 7 such का कुछ है क्या?
Your time is over. IISc, you have 3 minutes to present your answer. Your time starts now.
Okay, so for the first question, we have observed that in low IISER conditions, the mRNA transcript is in 7,000 bases in size, and in high IISER rich conditions, it's in 140 bases. So from the structures given in observation 4, we can infer that in low IISER conditions, it's in the form of it's in the second form, which is region 2 binds to 3. So therefore, the poly A tail, which is in the end, does not destabilize the DNA RNA configuration, so it can be transcribed fully. It produces 7,000 base pairs, which explains the observation 3. And in the first, the loop along with the poly A tail destabilizes the conformation. Therefore, it's in the 140 bases size. So yeah, this is I have seen observation 3.
Okay, observation 3. 1 minute 40 seconds. You explain everything above.
That's it.
Yes, the binding also prevents IISER from forming because these are the IISER codons. So this getting loop function also prevents IISER. And for in the IISER rich conditions, this formation, these loops forming stop the IISER codon from getting transcribed because the ribosome will just skip past them. So that prevents that from happening. So you don't produce more IISER, or you don't ask
You
for
have
more
1
IISER when you
minute
have a lot of it already, because IISER is expensive to make. And that doesn't happen here because while 3 may have IISER codons, these codons are free to get transcribed. So, and this continues on, and yeah, you did say the part where it goes to IISER Vienna. And because this is not getting destabilized, this continues on and allows the transcription and translation of either BNA, which
30
gives
seconds.
helps use the machinery for producing more IISER. This helps in making more IISER amino acids
It itself. also explains observation 2 why even though transcriptional regulation is going on, there's post-transcriptional regulation happening, which which allows the
Your time is over. IIT Madras, you have 2 minutes to present your answer, and your time starts now.
Okay, so for the first question, usually regulation occurs like there will be a promoter, operator, and on top of that a repressor, and like something like lack of operand comes, and like if lactose is there, that will go. In similar case, I'm considering IISER MB to be something like a repressor. I mean, my hypothesis is that IISER MB acts like a repressor, and in my model, if IISER is like if IISER is exists than required, then IISER MB will basically inhibit the regulation, which also proves in observation 2 why they say IISER is negative regulatory. And observation 3 states that short mRNA bases, many short mRNA bases of 140 bases form. I feel like that will happen in this particular scenario, and the 7,000 bases will form in this scenario. And for the second question, I feel like 2 successive IISER codons are required because when can you explain this first?
For the third question, we believe that quoting from observation 2 that a binding is chemically blocked in IISER rich conditions. So when my IISER will be rich, I need a dominant mechanism to
30
stop the
seconds
IISER
remaining.
production. That's why the second regulatory mechanism, which is the transcriptional regulation, will be more active in higher levels of IISER, and that is more dominant as compared to the binding complex being able to stop the translation. So for the second.
Okay, so I feel like when seven such successive codons are there, the regulation will be more efficient and more specific, and the accuracy will be more.
Your time is over. IISER Bhopal, you have 2 minutes to present your answer. And your time starts now.
So for the first question, we are hypothesizing that the region 1 binds with region 2 when it is IISER rich conditions, and the region 2 binds with region 3 when it is deficient condition. This is because when it is in the rich condition, the IISER codons won't be open for translation. That is why it won't need to it won't produce any other amino acids. And for the 2 and 3 binding, the 2 codons of the amino acid would be open for translation. That is why it would be more beneficial to form a loop there. For the second question, having 2 successive IISER codons would help the process being more efficient, because already the amino acids are relatively lower in concentrations in the environment. So that would help for faster production. Having 7 successive codons will be detrimental, I feel, because as the rate is faster of producing the codons, they will also start inhibiting the process. So 7 successive would produce a lot of amino acids at regular intervals. As for the third question, at mild to moderate levels of deprivation, there are amino acids present that could help in inhibiting the process. So using those would be more energetically favorable than going to a transcriptional regulation. Transcriptional regulation helps when there are like extreme levels of deprivation. So there is no amino acid binding to the blue molecule. That is why the bacterium chooses to go to the transcriptional regulation to inhibit the process. Thank you.
Judges, do you have any questions for the teams?
Missed how do you explain that 7 codons is better, or better for sensing IISER levels? And
No, I said 7 codons would because there are 7 regular intervals of codons for an amino acid. As it is produced, it will also start inhibiting the process. So it would be more efficient. Less efficient. Sorry. No, I think it is not a better sensor. Yes.
I had a question for the first team. So what was your answer for question 2 and 3? Did you have an answer? You had your question 2 answer was very brief, I think, and 3, I don't remember whether you said anything.
In the starting, it's an intuition that the reason you regulatory mechanisms is, for example, because we know it is very expensive to produce. And if it is very rare in the environment, then it's like more necessary to produce it, and you have a mechanism separately for that. And then if it's mild to moderate, you don't want the mechanism which produces it in extreme deprivation, because maybe just to make it faster, or whatever manner, it's only activated. You need to produce it, given that it is so. And as for question 2,
So in question 2, I just said that since amino acids are probably since the ISR is probably less in compared to other things. The two successive codons for its synthesis are given so that it is not skipped even in any chance. And in such cases where there are 7 successive codons, it will just ensure that the synthesis of that mRNA and subsequently the protein will always occur. It will just increase the probability of the synthesizing the proteins, the amino acids.
IT Delhi, you had mentioned that you had mentioned something about oh sorry Madras. You had mentioned something about it being a repressible operon, right? So could you elaborate on how you think it is a repressible operon?
So in the primer video you sent, I saw that it's like a promoter, operator, and on top of that the repressor. So basically, when this lacoperon, like this whole thing is, it's like a video. The example was about like lactose, like production of lactose and stuff. So in that they said if there is lactose present, it will go and attach itself to the repressor, and like if it will like take away the repressor away from the operator, and like the lactose will be produced properly. But if there is no lactose that is present, then we don't need lactose. So basically the regulation, that is what I explained. I took that as my mechanism and I did this.
But what you're explaining is for the lac operon. In this particular question, how do you logically arrive at the fact that this is a repressible operon? Since you made that argument, can you substantiate with information given to you in the question?
In place of lactose, we are like just that mechanism, IISER, like we're substituting IISER in the place of lactose, and like this IISER MB is what we are assuming it to be a repressor.
Thank you.
QMCs, I request you to explain the answer.
So to the IIT Delhi team, I did not understand your answer to the presence of 2 versus 7. Do you think 2 is 2 increases, 7 having 7 codons in tandem increases the
I believe that 2 is there can be an optimum number of codons between 2 and 7. So since at 2, I think it is it has increased the sensitivity by a certain amount. But if you keep on increasing it till 7, I believe there could be a decrease, because as more number of ISR are produced, it will subsequently decrease its own production, as we had initially been given that ISR in its own synthesis.
Matlab also that 2 places for and you have a moderately IISER deficient environment, then probably it will bind in one and stop the loop formation. But in 7 you will always have those free ones, like until and unless you have very high IISER concentration, you will always have free codons which can then form the loop. So it is more regular to be only 2 IISER.
I request a member of the question making team to come here and explain the answer.
It's fine. Huh? Yeah. 2 maybe? 2. All right. So we are looking at a regulatory system. Most of you did not come to the regulation part at all. So we'll touch upon it entirely. We'll look at it. Okay. So this is IISER Pashan. This is the MB region, and this is the what was it? BNR. So what we're saying is that this is the machine. These genes synthesize the machinery for synthesizing more ISR. All right. And when there is a lot of ISR, these things bind to a repressor, which inhibits the translation or transcription. It basically negatively inhibits the synthesis of the machinery that these genes are producing. So the question is, if this molecule is inhibited, how is this system still working? That is the question. And what we are looking at is that this region, the transcript of this region has region 1, 2, 3, and 4. 1 and 2 can complement with each other. 3 and 4 can complement with each other. 2 and 3 can also complement with each other. What is also important is that 1 has 2 successive IISER codons, and between region 1 and 2, there is a stop codon. All right? So now we'll look at how the dynamics play out when there is IISER rich conditions and when there are IISER deficient conditions. So we'll look at first how, so in IISER deficient conditions, also one important thing that I think none of you touched on was that this is a bacteria, and transcription and translation are simultaneously happening in the cell. That has been mentioned in the question. Okay, so when this transcript is being formed, the ribosome is moving on the transcript right behind the RNA polymerase. All right, so in IISER deficient conditions, tRNAs charged with IISER will also be deficient. All right, so when this region is getting formed, when the region 1 is getting formed, and the ribosome comes here, it does not find enough charged tRNA to place here. So it stalls there. It stops there. The mRNA polymerase, on the other hand, goes on transcribing the rest of the transcript. Right. So now region 1 is blocked. So region 1 can no longer bind to region 2, but region 2 is now free to bind to region 3. So there's a tiny kinetics playing out here. So the ribosome has to stall in region 1 for long enough for 2 and 3 to get transcribed, so that 2 and 3 forms this structure. And if this structure is formed, there is this poly A region followed by a there's this non-poly A region which is before this poly A region, and now this structure is not destabilized, which means the entire transcript can be formed. This is what is happening in IISER deficient condition when the ribosome is stalling in region 1. In IISER sufficient conditions, the ribosome does not stall in 1. So when 1 has been transcribed, the ribosome does not stall in 1. It follows the mRNA polymerase, comes to this stop codon, falls off. The mRNA polymerase, however, continues because it has nothing to do with the stop codon. Region 1 is formed, region 2 is formed, region 1 and 2 forms this structure, region 3 is formed, 4 is formed, region 3 and 4 forms this structure. This loop is immediately followed by a string of A's, which destabilizes this structure. The transcript does not form anymore in this case. So in this case, you have the transcript getting formed. In this case, the transcript of the genes we want to synthesize are not even getting formed in the first place. So if there's no transcript, there are no proteins. So this is the regulation we are talking about. Okay, what was the second part? Yes. Yeah, the second part talks about why we need to have 2. So 2 is just like a random number
It could be more than 2 as well. It just, so biological systems are subject to noise, right? So 2 just reduces, it just makes the system more as to any noise, because IISER is already a very rare amino acid. It is much more probable, it is much less probable to find 2 successive IISER charged tRNAs as opposed to 1 IISER charged tRNA. So that is entirely what 2 does here. What happens if you have 7? So when you have 7, of course, the sensitivity increased with the same logic I just said. What also happens, what we can infer from a system having 7 amino acid, is that you really do not want to produce this amino acid after a high concentration. Okay, why? Because perhaps that concentration is lethal to the cell. So this is like an open-ended question. We are not really expecting a particular answer to that. The last question is, why do we have two mechanisms in the first place? So, okay, so if you look at the 2 mechanisms are of course the repressor-based mechanism and the mRNA-based mechanism. So the repressor-based mechanism is an indirect proxy of how much IISER is there in the cell, because the repressor binds to IISER. And that is what is inhibiting the synthesis of IISER subsequently. So the repressor bound to IISER becomes the signal and not the IISER in the cell itself. In this case, however, we are looking at the direct charged tRNA, IISER charged tRNA in the cell, which is a much more direct signal than this. So this is a much more sensitive, fine-tuned control of the system. So that is the answer. Any questions? No, that's it. Yes. It is a tRNA operon. But do you not just have which one?
Thank you, Soumya Jyoti. Team IISc, you have the choice between 3 questions: united we stand, divided we photosynthesize. This is a small question and machine learning.
This is a small question.
This is a small question. Scientist P found an alien organism which shows an interesting excretory system. The system has the following parts. Blood is filtered by the filtration unit, and the resulting fluid, which contains urea, water, and ions, no proteins, enters the hairpin. The final urine, which is to be excreted, comes out from the collection system. The arrows indicate the direction of fluid flow. The interstitium is made of extracellular matrix and shows an osmolarity gradient as is shown in the figure. This is figure 1A and figure 1B. Region A of the hairpin is freely permeable to water but impermeable to ions. Region C is impermeable to water but has ion transporters. Region B is impermeable to both. P identifies 3 types of ion transporters in region C, which are uniformly distributed along the region. This is figure 2. M plus N plus ATPase is the only ion transporter that uses ATP, the energy currency of the cell, for this process to transport the ions against their concentration gradient. MnCC transports the other 2 ions, N plus and C minus, in the same direction as it is transporting M plus, irrespective of the concentration of N plus and C minus. Also note that MNCC works only when all the ions that it transports are bound to it. To study how the osmolarity gradient in the interstitium is established, P isolates only the hairpin part of the system along with the interstitium. They pass some fluid through the hairpin, same in composition as the one that passes through it naturally, with an osmolarity of 300 units. They repeat these for multiple cycles. Initially, the osmolarity is 300 units across the entire interstitium. The results are depicted in figure 3. This is figure 3. All cells in the system have the same higher concentration of N plus and lower concentration of M plus and C minus as compared to any extracellular substance. Both the interstitium and the fluid in the hairpin count as extracellular substances. Questions. 1. When P created a computational model with only the known ion transporters, the gradient formed was much weaker than what was observed experimentally. Also, the electrical potential of the lumen was positive in the actual experiment, while their model predicted it to be 0. What additional transporter would you expect to find in the system, and where would it be located? Explain. Question 2. The osmolarity of the fluid entering the hairpin is always 300 units, and at the start of the first cycle, the concentration of the ions in the fluid in the hairpin and the interstitium is the same. A. Explain how the gradient is established and maintained for the first cycle, 3A to 3B. Comment on MNCC's features with respect to its speed and efficiency. B. What facilitates the transition of the system from 3B to 3C? Question 3. What will be the osmolarity of the fluid exiting region C of the hairpin compared to the fluid entering region A in the first cycle of the experiment and in any subsequent cycles? Provide appropriate explanations. Question 4. The interstitium needs to be supplied with oxygen by the blood vessels. It was seen that this organism has special blood vessels which are freely permeable to ions and water and run along the hairpin as shown in figure 4. The blood entering and exiting the interstitium through the blood vessels is the same osmolarity, which is 300 units. This U-shaped arrangement of blood vessels is needed for preventing the established osmolarity gradient from being disturbed by the blood flow. Why do you think this is the case? Comment on the direction and speed of blood flow through these blood vessels. Question 5. The hairpin connects to the collecting system, figure 1A, which is responsible for concentrating the urine. The interstitium absorbs both water and urea passively from the collecting system. Why is the absorption of urea necessary in this case? That is it. Participants, if you have any doubts, you can ask them now.
What is meant by osmolarity here?
You can ask a question.
What is meant by osmolarity?
No, it's not molarity. You're supposed to know. You are supposed to know what osmolarity is. All right. Do all of you require a clarification on what osmolarity is? It's a. Okay. So it's a measure of the concentration. It's different polarity. Osmolarity really depends upon the total number of ions that are present in a particular fluid. So instead of really looking at the specifics of osmolarity, if you remember from property, it depends on the total number of ions and not on the individual identity of the ions.
But independent of, like, form of molarity, but independent of the ion. Okay.
It's a measure of the concentration.
If there are no more doubts, teams, your 15 minutes of thinking starts now. Participants, you have 10 minutes remaining. Participants, you have 5 minutes remaining.
Exactly, yeah. I think this is completely I don't. I'm ignorant. I don't. Oh. This one.
Participants, you have 1 minute remaining.
All the questions make my head spin. Mathematics. Mathematics gave me zero. Mathematics got
30 seconds remaining. Teams, your time is over. IISc, you have 5 minutes to present your answer and your time starts now.
For question 1, clearly there must be some other ion transporter in the cells of region C that has not been considered here, because when as the computational model is going against the actual experimental results. Now the points in the question say that the firstly, the gradient form was much weaker than what was observed experimentally. Like theoretically, it was much less experimentally, and the electrical potential of the lumen was positive, while the model predicted it to be 0. So, if we look at the transporters in region C that are interacting with the lumen, it's only the MNCC which is transporting M plus in this direction, because M plus is higher outside the cell, as I was stated, is higher in the interstitial, in the lumen, and therefore N plus and 2C negative are also getting transported here. Oops. Here. And if you look at the charge distribution, this is affecting, like there are 2 positive charges and 1 negative charge is moving here, so it's leaving the lumen neutral. But the lumen is supposed to be, is expected to be positive, and so we are expecting another, what is it called? Ion transporter, an N plus transporter. This is not going to be using any ATP. As it said, there are no other transporters that use ATP apart from the M plus N plus 1. So there must be another N plus transporter, because we must be providing some positive ions to the lumen itself, and the only way to do that is because the concentration of N plus inside the cell is higher, so there must be an N plus transporter that is not being considered, that is transporting the ions, N plus ions from the cell into the lumen, which is making it more positively charged.
So for the first question, it's given that there's a blood vessel which is flowing parallel to this hairpin loop. It is parallel to this hairpin loop because if it is flowing in any other way, it might like absorb water like ions like randomly, because it's freely permeable, and it might disturb the osmolarity gradient. So when it's parallel, I'm proposing that it will take up ions as it's the blood is flowing this way, by the way, opposite to the flow of the filtrate. So as it's flowing opposite to the flow of the filtrate, it's absorbing some ions, which is helping to produce the osmolarity gradient, and as it's flowing down, it releases some ions here. So this might help produce the gradient, and it was given in the question that in the fifth question, it's given that collecting system releases some urea. The interstitium absorbs some urea from the collecting system. This will most probably be due to it's absorbing it here because this increases the osmolarity here, and it helps produce osmolarity gradient, and so it can absorb water as it's flowing, as the filtrate is flowing down, to concentrate the filtrate even more. So yeah.
Okay, so as we're going downwards, we know that N plus concentration is the N plus concentration is less, so the MNCC, it is said that it will require all N plus, and C minus to bind to it to operate. So that will not be able to operate, so the dominant mechanism will be the N plus mechanism given.
You
over
have
here.
1 minute remaining.
That's it. Okay. It is possible that the first cycle of the gradient might be arising because simply of the fact that as ions are being pushed in sort of uniformly to the intracellular medium, but this is from where the ions are exiting, so that is why there is a gradient. But
IIT Madras, you have 3 minutes to present your answer.
Yeah, so
Starts now.
hello. So we have M plus N plus ATP as the only gate which transports that uses ATP. That's because of the concentration gradient being the reverse to what it was transported. And irrespective of the concentration of N plus and C minus, so the MNCC transports all the ions in the direction it's transporting N plus. So for the first question, we are given that the gradient formed by like scientist P was much weaker than that which was observed experimentally. That's because since the osmolarity depends only on the number of ions, that means the number of gates which scientist P has produced, made in his models are less. And we need to have a gate which transports an ion into the which transports some ions such that the gradient becomes larger. And in diagram 1b we can see that in diagram 1a and 1b combined, we can see that the descending loop is ion impermeable. That's why it doesn't contribute to the osmolarity, while the ascending loop will have ion gates. So for the first question, the answer is that the N plus transporter will be in the boundary of the interstitium and in the boundary of the cell and the lumen, such that the lumen becomes positively charged. Since M plus, N plus and 2C minus balance each other, we need a positive ion transporter such that it transports from the intracellular material to the lumen. And since N plus is higher concentration in the cell, we have an N plus gate. For question third, before the first cycle, the osmolarity is same, but in the ascending loop, after one cycle, we'll have N plus ions transporting into the lumen. So that's why the osmolarity will increase for the bottom parts, and as the ascending loop progresses, the osmolarity will see a gradient. But after 7 cycles, this system stabilizes to what resembling to an original system.
So for fourth question, they said that the blood vessel parallelly comes and supplies oxygen to interstitium here. So I feel like this is necessary because this is the case.
30 seconds.
I feel like this is the case because if again, like IISc said, if it's in any other direction, it will directly affect other stuff. Whereas because it's parallel, it will just take away some ions. I just want to add on to the fact that because it's taking away some ions, I feel like the velocity will reduce. The blood velocity will reduce, and the direction will go from this side to this side. Is what I feel like would happen.
Time is over. Your time is over. IISER Bhopal, you have 2 minutes to present your answer and your time starts now.
For the first question, the additional transporter that would be there would be the one to maintain the N plus concentration in the C cells, the C region cells, and also the ATPase, they work to diffuse the M plus and N plus. So the N plus is actually already concentrating a lot within the cells. So to balance that out, some of the N plus will remain in the region of the lumen. Then for the question 2b part, to attain the optimum gradient, what facilitates the transition of the system from 3b to 3c would be the osmotic pressure. And for the fourth question, the direction would be that to the opposite of the U-pin loop, because some of the...
You have 1 minute remaining.
Some of the ions are getting reabsorbed as well in the blood. And for the fifth question, the reabsorption of urea is necessary even to maintain the osmolarity and osmotic pressure of the cells. So some of the urea is retained in the interstitium and not all of the urea is excreted. And if that is not the case, some of the urea from the blood may enter the interstitium.
30 seconds.
So the water and the urea balance has to be maintained within the interstitium between and the collecting duct.
Please wait for the timer to run out. IIT Delhi, you have 2 minutes to present your answer. And your time starts now.
Yeah, so our model for our extra transporter which is required is that we have a transporter in the cells towards the lumen, which is taking M plus from here to here. Yeah. नहीं, एक छोटा सा देख दो ना. So because we know that like n plus is higher, n plus is higher inside the cells than it is outside the cells, we propose that the way the ion gradient is working is that n plus is flowing outwards, because of which m plus is. Well, what we need to explain is that there is higher molarity outside the cell wall than there is inside the cell wall. So if we are able to prove that there are more ions going out, then it is in our favor. Now over here there are 3 going out and 2 coming in, so an extra 1, and similar for C minus, which is why osmolarity is decreasing, increasing over here and decreasing over here relative to that, because it's equal, or there are those going inside. And
for the fourth question, as we know that blood is coming at 300 osmolarity,
30 seconds.
and when it reaches the bottom part at about when the interstitium has at about 1200 molarity, due to higher pressure, it will clearly slow down, and hence the velocity of blood will be higher at the when it will be entering, and lower when the pressure of the interstitium is the highest. As for the fifth question, we can say that urea has.
Your time is over. Judges, do you have any questions?
Them all answered. So, can you explain the ISC? Why do you need urea to be absorbed? I mean, the question 5. Can you just clarify that a little bit?
So urea is absorbed at the bottom of the collecting duct from the collecting system. This increases the osmolarity below to maintain the gradient and basically facilitate the absorption of water again to concentrate the urine.
If you absorb water, you'll be concentrating the urine, or?
Like the water is being absorbed from the filtrate. So urea is basically absorbed out of the filtrate into the interstitium so that the concentration is higher in the bottom.
Okay. Okay.
Are there no more questions? I request a member of the.
Question is to IISC. You talked about, so you mentioned that the direction must be opposite to the direction of blood flow must be opposite to the direction of flow in the lumen.
Yeah.
Why? What is the issue? Like, can you say why?
So if the blood flow flows in this any other direction, it'll basically it's freely permeable, right? So it'll absorb like ions or anything along with the osmolarity gradient, which will mess up the gradient basically.
Okay. And I had another clarification. In question 2, you were talking about some dominant mechanism here. Can you substantiate on that?
No, it is just a possibility that the MNCC, it is mentioned that it requires all the ions bonded to it. So if we're going downwards, we have a less amount of M plus. So that is why the MNCC might not be the main factor in transporting the ions out, and it might just be the N plus is happening.
All right.
I request a member of the question making team to explain their answer.
Give me a chunk. Yes. Am I audible or do you? All right, yeah. So I think that those were pretty good attempts. The first question most of you got correctly, so I'm not going to go into that in the interest of time. So the second question, essentially what we were expecting is, consider the first cycle. In the first cycle, 300 units osmolarity fluid is entering and your interstitium is also 300 currently everywhere. So in the first cycle, and this region is permeable to water but not to ions, this region is impermeable to water but it has specific ion transporters. So in the first cycle, what's happening is, when your fluid is coming down, there's no loss of water here that's taking place, but there will be loss of ions that will be taking place here. And now if you did the charge balance and if you worked out the direction of the ion movement, you would have realized that there is a net efflux of M plus and C minus ions out into the interstitium from the lumen. So what essentially effectively is happening is, as you move upward here, you are putting out this M plus and C minus ions. Now that still does not really explain how the gradient is formed. How the gradient is formed is that when your fluid is reaching the bottom layer, it has a high concentration of these ions. Your MNCCs will work more, they will put more of these ions into the cell, and then the cell has other transporters on the interstitial side, which will again put out these ions. So effectively what is happening is that since there is more ions in the fluid here, more ions are being put out in the interstitium, but by the time the fluid is reaching here, the ion concentration in the fluid itself is decreasing, which is why you will have less ions here. This explains what is happening in the first cycle. In the subsequent cycles now, there needs to be another parameter to ensure that not only is this gradient maintained, but this gradient has to be steepened with each cycle. Here, what we were expecting is the direction of ion transport that is happening from the lumen into the interstitium horizontally. This rate of transport has to be much faster than the rate of diffusion that is happening in the interstitium for a particular horizontal level, which would maintain the gradient and which would further concentrate the interstitium and steepen the gradient. That was what we were expecting for the second question. In the third question, the osmolarity of the fluid that is coming out of here will always be less. And why is this so? For the first cycle, it's pretty easy to understand, right? For the first cycle, this is 300. There's no water movement here because it is also 300 in the first cycle everywhere. Here, there is ion movement. So from the fluid, you're really just throwing out ions. So effectively, here, whatever you'll get will be of a lower osmolarity. However, in the subsequent cycles, once you have established some sort of a gradient here, yes, you will throw out water here, but do remember that we have mentioned that the cell is just permeable to water, which means that this transport of water will happen through diffusion.
there are no special transporters who are actively transporting this water out, which is why the transport of this water will be slower as compared to the transport of these ions, which essentially means that again by the time you reach here, the osmolarity of the fluid will be lesser than what the osmolarity of the fluid that was coming in. For the fourth question, yes, the direction of movement, again, quite a few of you got it right. The direction of movement, so if this is the direction of movement of the fluid in the lumen, the direction of blood flow has to be opposite. The main function of this blood vessels are to ensure that, so blood is necessary to oxygenate the interstitium. Now you have to ensure that when blood is going in, it doesn't wash out the gradient. What this ensures is whatever ions are being taken in when the blood is going down, those ions are being again put out when the blood is going up, thereby maintaining the net ion concentration and thereby the osmolarity gradient in this interstitium. What the issue that would happen, and this is a very fine point, that if you had the blood flow from the other direction instead, is that if you have the blood flow from this direction, remember this region is permeable to water but not to ions. Essentially what that would lead you to do is you are taking up ions from a region that's not permeable to ions, and you are taking up water from a region that's not permeable to water. You would end up setting a sort of horizontal gradient, which again could potentially disturb this vertical gradient that you have. For the, so the speed. The speed of blood flow needs to be slow enough so that the diffusive processes can occur. Remember, the blood flow through the blood vessels that are occurring and the exchange that is happening, that is based on diffusion. And for diffusion to occur, the flow needs to be reasonably slow. It can't be too fast so that diffusion, there is not enough time for the diffusive processes to occur. For the fifth question, yes, so I think one of you guys, I think someone mentioned, I forget which team, but that was pretty much correct that yes, urea and water have to be absorbed to basically maintain the osmotic gradient that is here. So if you just keep on taking up water, you'll end up diluting this region. And then what that means is that not only are you disturbing this concentration gradient, but the whole role of this concentration gradient, which is to concentrate the urine, is no longer fulfilled, because once water moves in here and this is dilute, the urine cannot get concentrated further as it is moving down the collecting duct. Yeah.
Thank you, Soumya Jyoti. IIT Madras, you get to pick the next question. Your choices are
Yeah, a general note to all the teams, as you might have been briefed earlier, it is recommended that if you are not the team to whom the question was directed to, that you add, you don't try repeating the same answer that previous teams have said, and instead spend your time on either adding on to the new question, or if you feel that the teams that have come before you have said something wrong, refute that and justify that statement. If you feel whatever the team that said before is correct, you don't need to repeat that. Either you refute that statement and justify why, or you add on something to the question, but try not to repeat the same things.
Your choices are between United We Stand, Divided We Photosynthesize, and machine learning.
United we stand, divided we photosynthesize.
United we stand, divided we photosynthesize. Professor Vakula, a plant biologist, was studying the genes, namely Hem-A, Hem-B, Hem-C, Hem-D, and Hem-E, involved in the division of chloroplast. The schematic shows the division of chloroplast. In a wild-type cell, chloroplast division begins with one protoplastid. In phase A, it divides 4 times, and in each division, a single protoplastid grows in size and then divides into 2 protoplastids. There are 16 protoplastids present at the end of these divisions. Phase A takes k days to complete, after which the existing protoplasts immediately proceed to phase B, irrespective of their current size. In phase B, the chloroplasts undergo 3 divisions, wherein each division, the chloroplast first grows in size, forms a furrow that gives it a dumbbell-shaped structure, and then divides into two new chloroplasts. Note, a successful division can occur only if chloroplasts have reached a specific optimal size. Once a chloroplast has reached this size, further growth is stopped by one of the 5 genes. Chloroplasts of other sizes cannot divide. During division, each chloroplast has a plane of division. If it is located in the center of the parent chloroplast, the resulting daughter chloroplast will be of the same size and of different sizes if the plane is off center. These 3 divisions take m days and result in 128 chloroplasts, which continue to grow in size. With the intention of constructing the pathway using 5 genes and without much regard for the social life of her students, Professor Vakula makes her students culture single and double null mutant lines for each of the 5 genes. A null mutant for a gene does not produce the functional proteins coded by that gene. Single null mutant does not produce only one such protein, whereas double null mutant does not produce 2. The students create 15 null mutants, 5 single and 10 double. All the mutations are recessive. All the mutations are recessive in nuclear DNA. Assume no sex-linked inheritance. The students grow all 15 lines for K plus M days and make the following observations. Professor Vakula knows parts of the pathway that are regulated by the 5 genes but does not know how the exact positions of each 5 genes in the pathway. Help her out, won't you? These are the observations made by the students. Question 1. Where in the pathway do you think hem D should be located relative to hem B, hem C and hem E? Likewise, where do you think hem B and hem C lie with respect to each other? Question 2. What functions do you think 4 genes hem B, hem C, hem D and hem E are performing in the pathway? Place these 4 genes in 5 boxes. Obviously, there will be a repetition, and explain the phenotypes obtained in each case. Question 3. How and where do you think hem A functions? Question 4. Deduce what effect the hem E mutant is having on the chloroplast division. One of the mutant genes, hem E or hem B, partially overrides the effect of the other's mutated state on chloroplast number and division. Which one is it and what is being overridden? This is the end of the question. I would like to ask the participants if they have any doubts regarding the questions. No question?
Can you clarify and place these 4 genes in 5 boxes and explain the phenotypes? Okay. 5 boxes means consecutively, right?
This schematic 1, 2, 3, 4,
okay,
5.
okay.
Yeah.
Yeah, fine.
Any more questions? No questions?
All right. So question 3 will be how and where hem A is placed in the pathway, and not just where or how, whatever. You have to answer both.
Okay. So no further questions. So, do you have another question? The thinking time starts now. Less than 5 minutes remaining.
So we have 16 from this cycle. Right. We have 16 proclastic from this cycle. Yes. And if they have a doubt, I'll be back on. So if the only if it's asymmetrically positioned, will it have variable sizes? So this gene is positioning the controlling the position of the division apparatus. We have two equal sides, three sides. No, we have the same size. We have mentioned it in the paragraph that wild type cells have the same size. So we are showing that this both things can happen. That has been mentioned in the paragraph that if the division is like happening symmetrically, two sizes will be produced, corresponding, and if it is not if it is symmetrically dividing, there are two same sizes. In the drawing, it looks like one becomes this, one becomes not necessarily.
Only 1 minute remaining.
Whatever is there is referred to as a chloro.
Participants, you have 30 seconds remaining. Time's up. IIT Madras, you have 5 minutes to present your answer. And your time starts now.
So for the okay. So in the first question, we are asked where in the pathway HEMD should be located related to HEMD, HEMC, and HEMI. So when we have two mutants, we can see HEMD, HEMD is identical to HEMD, HEMC, HEMD is also identical, and HEMI, HEMD is also identical. So this shows that HEMD should come before all three of them. Also, it makes a very the global size is very large, so the one should be HEMD. And similarly, what we can see is in HEMD and HEMC, we get identical to HEMD, so HEMD should come before HEMC, and similarly, HEMC should come before HEMI. Okay. In the second question, we have to place HEMD, HEMC, HEMD, HEMI. So,
So with similar arguments, we can see the additional information where we have variable chloroplast sizes for mutants which are hemi mutants. So basically, when hemi is absent, the mutant hemi protein is absent, then it leads to an asymmetric division as compared to a symmetric division. So hemi कहाँ पे? So
HEMD, क्या करना है अपने को? क्योंकि HEMC, HEMI इफेक्टिव, बट HEMI देखो, HEMI ये सब asymmetric division है. ये कर दो. तुम सोचो फिर. नहीं मैंने ये सी ये डी, क्योंकि E का मतलब है asymmetric division. यहाँ लिखा है कि vectors everything HEMD and HEMC lie with respect to each other. तो ये कुछ कर ही नहीं रहा. देखो ना HEMD और HEMC का ऐसा है कि HEMD आएगा. ये कौन सा होगा? क्या बोल रहा? ये because of asymmetry. हाँ.
Yeah, so due to variable chloroplast size, Hemi decides if Hemi is there is an Hemi mutant, then more asymmetric division is there. And it decides whether it's symmetric or asymmetric. And Hemi, as we can see, only one chloroplast is present when when Hemi is not present. So in Hemi mutants, so that's why Hemi should block the division. That's why Hemi should be in box number 1 and 5. In box number 4 is the selector bit, that's why 4 has Hemi. And in case of HEMC mutants, we can see that mostly we have dumbbell shape chloroplast. That may be because of two cases. The first case being the phase 1 where we have a dumbbell shape. The second case being in phase B where we have a dumbbell shape due to a symmetric division. So when we see HEMC combined with, when we see double line mutants, HEMC, Hemi, HEMD, HEMC, we can see that they are HEMD, HEMC is identical to HEMB mutants. So that may be due to the chloroplasts which are in phase A, the dumbbell shaped chloroplast. And for HEMC, Hemi mutants, we have a 68 number of chloroplasts, which means that the phase A has been completed and phase B has not been completed to the full. So and by the numbers of HEMD, HEMC being 16, we can say that HEMD, HEMC have dumbbell shaped mutants, dumbbell shaped chloroplast due to only phase A the chloroplasts. So
30 seconds remaining.
Yeah. So HEMD mutants have 10 number of chloroplasts in place of 1. That means Hemi is functioning along with HEMD.
So I feel what is this Hemi is affecting the asymmetric and symmetric dumbbell shape in a different way. That's why we are getting 96 96.
Time's up, guys. Time's up. Next, IISER Bhopal, I request you to present your answer within 3 minutes, and your time starts now.
Okay, for the first question, where in the pathway do you think hem D should be located relative to hem B, hem C, and hem E? Okay. So in the pathway, I think the hem D would be located in the box 1, since it is promoting cytokinesis. And when we are removing the hem D, we have only one cell that is very large. So this is the one cell which we are obtaining, and it is very large. Then hem B and hem C, with respect to each other, lie in the box 2 and 5. Now, the C lies in the box 2 is because when we are removing C, we are getting a very large cell. So this is again the very large cell which we are getting, and then it is not undergoing the further, and then we are getting dumbbell shape. So we are getting this kind of cell instead of this kind of cell when we are inhibiting C. So if C were present, we would get this kind of cell. Okay, where in the pathway do you think hem A functions? So in the pathway, we think A would function here for the fourth. And why is it so? It is because A is the one which is controlling the cell growth. So after a certain point of time, this cell is not going further. And that is why the A would be placed there. And D, C, E, हाँ B हो गया ना? ये कुछ है?
30 seconds remaining. Done. I request to reset the timer for IIT Delhi. IIT Delhi, you have 2 minutes to present, and your time starts now.
Okay, so as per us, the first one will be hem D, the second will be hem B, third will be hem E, fourth will be hem E, and fifth will be hem C. So hem D is the one carrying out the first process, that is the division of the protoplast, which is why in the absence of hem D, there is only one available. And hem B is the one that's causing the growth of the protoplast, so that of the division of this molecules that is of appropriate size for further division. And the fourth one is hem E because it allows it in the presence of hem E we have symmetric breaking, in the absence of hem E we have asymmetric breaking, and hem C is the one that performs 5, that is after like it breaks down the dumbbell into 2 molecules. Now for the repetition it's hem E, and for the fourth one which is overridden, so in the absence of hem E, hem B gets overridden, as in the absence of hem E, even hem B is not able to show its phenotype, which is meaning that in the absence of hem E and hem B doesn't function, which can be seen in the fact that in the absence of hem E we have 32, and all the given information is same as in the absence of hem B and hem E.
30 seconds remaining.
And hem A, okay, so in the absence of hem A, we can see an increase in the number of chloroplasts, which means that hem A restricts the growth, and in the absence of hem A, growth is more. Yeah. Number of chloroplasts is more.
The absence of hem A means that the phase 1 is able to happen more times, so you have a lot of these chloroplasts, which...
Time's up, guys.
Yeah.
I see you have 2 minutes to present your answer, and your time starts now.
Okay, so for the fourth question, I'm proposing that Hemi affects the fifth division because it's producing 32, and after that it's not affecting. I mean, after that it's not dividing, so it's affecting the fifth division of the chloroplast, and it's providing two populations with different size. So before that division, I'm proposing that it's affecting the like dumbbell formation, and it's producing an asymmetric dumbbell. The mutation of E, it's responsible for the symmetric formation of the dumbbells. When it's mutated, it produces an asymmetric dumbbell. So that's why there are two populations with different sizes, and E overrides the function of HEMD because when both are mutated, HEMI and MD, it still produces 32, but it's only partially overridden since two populations are produced, both larger, so it's only partially overridden.
About Hemi, so there are two ideas that we have. One idea is that Hemi is so what is happening is we can observe that for Hemi we have 10. For Hemi we have 10. So that might suggest that when we reach this point, before the action of Hemi occurs, before the action of Hemi and the other things occur, Hemi is able to split these. So even if HEMD is blocked, you just have this, which is a protoplast, and Hemi is able to act on that. And if half of them will give us 8 into 8, 64, and 8 into 2, 64.
Time for guys. I'd like to ask the judges if they have any questions for the
So, I think the question about HEMA, what is HEMA doing, right? Can all of you quickly repeat what you said HEMA was doing? I think maybe you didn't get to that question at all, right? Or did you get to that question? HEMA, what is HEMA doing?
Yeah, I just.
What was your answer for that?
I was just guessing that Heme might affect this, might treat asymmetric and symmetric different dumbbell differently.
Okay.
And so that's where we are getting.
Okay, and you?
According to us, the Heme is controlling the cell growth. So after a certain point, it is not allowing the cell to grow, and then further it is going under the dumbbell formation. So.
Okay. And you? You had something to say about Heme?
Yeah. Can you repeat? We have to repeat Heme, right?
Yes.
Yeah. So Heme causes, Heme restricts the growth. We can see that because in the absence of Heme, even the ones which were having less number of chloroplasts, in the absence of Heme, they have massively more number of chloroplasts. So Heme was restricting the growth.
But restricting the growth in any particular phase, or?
We think in phase B.
Phase B, okay. And
Okay, so as I mentioned, there were two ideas. So one idea is that at the end of the first phase, we'll have total 16 protoplasts. So what can, on the protoplast itself, what will happen is Heme will act and it will give us, like in the whole thing, it will give us, like say say it is happening normally and there's hem A, so you have 8 into 432 over there, and like if it is competing and it's dividing equally, and 8 into 16 will just follow the normal mechanism and give us 64. But if the normal mechanism is blocked because you're just forming protoplasts and you have just one, then only hem A will occur. Then the exact number 10 we are not able to explain, but because it is a protoplast, hem A can act on it and the number will increase. So we are able to qualitatively explain MDMA and the end of the it is acting. MD. MD.
So no. So in the phase 1 or phase B?
Huh?
Yeah, hello, no.
First phase. Yeah.
Hello.
Any more questions?
Yeah, hello. Can you hear me? Yeah. Could you tell what you said about hem A again? Like how are you like positioning it?
So if hem D is blocked, the other hem B, hem C, and hem E will not be able to operate because they're stuck at a protoplast. But hem A, so essentially they mentioned that after the hem D divisioning happens, we can consider them to be chloroplasts. I'm saying that hem A can act on protoplast itself. So what is happening over here is hem A will compete compete, and say they're competing equally. So you'll get 8 that will go on in the normal cycle, and you'll get 8 that will go on with hem A. So on the basis of that, I'm explaining, and yeah.
Thank you.
Oh yeah.
No more questions? I would like to request Somajyoti to explain the answer.
Hello. Am I audible? Okay. So, hello, can you hear me? Hello. Yeah. Okay. So, I'll just draw the pathway, that is easier. All right. So we'll look at the mutants and that's for this. So if we look at the hem E mutants, there are two populations. So clearly the segregation is happening, but so clearly the segregation is happening, but there has been a asymmetric positioning of the division plane, which is why we are having two different population size. This is the step where the where the plane is being positioned. So this is where hem E will be. If you look at hem C occurs here because if you mutate this won't be produced. So you have like large dumbbell-shaped chloroplasts. Yeah, hem B is what is controlling the size of the chloroplasts, and it is not letting the chloroplast grow too big when it is present. When it is not present, therefore, the chloroplasts are larger. If you look at the single mutant, there are 16 large chloroplasts, which means this division has happened, but after this, the whatever this thing was, that grew to an abnormally large size, which prevented division. So hem B is acting on this step and preventing this from being abnormally large. Hem D is into. Okay, so we are not really penalizing you for exchanging 2, 3, and 4 because the question, information in the question is not sufficient to position these 3 very well. So it's all in the same process basically. So hem D and hem D, what it is doing is that it initiates the proplastid and the chloroplast division. Therefore, so if hem D were just here and not there, you would have one proplastid at the end of the cycle, but this division would still continue fine. So at the end, you would have 2 to the power 3, so 8 to the power 3, 8 chloroplasts at the end. But if you look at the single mutants of hem D, you see there's just one very large chloroplast. So clearly it has to be positioned in 2 places. That's hem D. Hem A, on the other hand, if you look at the double mutants of hem A, all of them are intermediate. All of them have an intermediate phenotype. So that kind of hints to the fact that there can be an alternate pathway which is regulating this. And if you look at it closely, the alternate pathway has to occur in the proplastid pathway and not in the chloroplast pathway, because if it were in the chloroplast pathway, then it would then it would mess up with the numbers. If you look at it carefully, I really do not have time to do this, but if you look at it carefully, you should do the math when you're like not free, when you're free, sorry. So it really occurs in this pathway. That is the answer. Any questions?
Thank you so much, Jyoti. Let's proceed with the final question of this deep thought session, which is machine learning for IISER Bhopal. Machine learning. Following its discovery in Mimamsa 2024, Joe the fly becomes an attractive model organism for neuroscientists to study olfaction, the sense of smell. Neurobiologist Suhani is investigating the neural system that helps Joe smell, discriminate between, and learn new odors. She finds multiple layers of neurons and sets out to characterize their connections. This is the schematic of Joe's neural circuitry. The sensory layer comprises 50 types of sensory neurons that express a single kind of odorant receptor each. A single odorant can activate multiple neuron types to varying degrees. The activity of any sensory neuron increases with the concentration of the odorant. The second layer, labeled as the DJ layer, is made of 50 nodal structures, each corresponding to a sensory neuron type, and consists of 3 types of neurons. Type 1, VS neurons, there are 50 of them, each receiving direct excitatory projections from sensory neurons of the same type. Activity in the VS neuron increases with input from the sensory neurons and saturates after a point. Type 2, SP global, receives excitatory inputs from all the sensory neurons. Their output is directed to the junctions where all the sensory neurons meet the VS neurons. When active, SP global neurons reduce the intensity of the input entering the VS from sensory neurons, and this reduction is proportional to the total input received from the sensory neurons. SP local receive excitatory inputs from a subset of sensory neuron types that share similar odor response patterns. They project to the VS neurons of the same subset. When active, the SP local neurons dampen the activity of VS neurons. Note that there is a small but non-zero chance of any neuron misfiring. Question 1. When the SP global neurons were artificially blocked, Suhani observed that Joe could not process odors that were presented to it at high concentrations.
Provide a mechanistic explanation for this observation. Question 2. Explain mechanistically how the DJ layer processes two similar yet distinct odors. The NG layer consists of 2000 neurons. Each NG neuron requires the same threshold of input activity to fire. Odors are learned as activity patterns in the NG neurons which have projections to units that elicit different behaviors. The GRN is a giant non-spiking inhibitory neuron that receives excitatory input projections from all NGs and inhibitory projections to all the junctions where VS neurons meet NG neurons. Like SP global, activity in the GRN can dampen the input from VS to NG, and this activity is proportional to the total input the GRN receives. Suhani observes that each NG neuron receives inputs from approximately 7 nodes in the DJ layer. By looking at the structure across multiple flies, she also makes the observation that DJ to NG connections are completely random. That is, each NG neuron randomly samples 7 nodes from the DJ. Question 3. Propose an explanation for why the expansion in the number of neurons from DJ to NG layer is advantageous. What if the engineer also had 50 neurons? Question 4. Bit A. Provide a qualitative explanation for why all NGs take only 7 inputs from the DJ. Why not significantly more or less? B. Furthermore, what advantage does the random wiring provide over a preferential mapping where each NG connects to a specific DJ? C. What role is GRN playing in modulating activity in the NG layer, and what functional advantage is it providing to the system? What would happen if the GRN inhibition was constant rather than being proportional to the input from the NGs? This is the end of the question. I would like to ask the participants if they have any questions or doubts. No doubts?
So, hello. So the question says the GRN is a giant non-spiking inhibitory neuron. Can you clarify what non-spiking means in this context?
I mean, so it doesn't like, so neurons when you give them input, they sort of like have an increase in activity. And this is more like it provides like a gradual. Sorry, yeah. So a non-spiking neuron here means that it sort of, instead of providing like spikes of activity, it provides sort of a gradual continuous input that depends proportional to the activity it receives. Any other doubts? Okay.
The thinking time starts now.
Only 10 minutes remaining. Six minutes, 7 seconds remaining. 1 minute remaining. Less than 30 seconds remaining. Time's up. IISER Bhopal, you have 5 minutes to present your answer. Your time starts now.
Hello. First, I will explain the answer for 4B. The question says, what advantage does the random wiring provide over a preferential mapping? The random wiring would assess the different order, help to assess the different orders properly without creating creating a biasness. Also, I think the answer for 3 would also be similar, because having a larger number of NG layers would cover 7 different nodes equally and would be more efficient to detect the difference between 2 orders.
For 4A, why provide a qualitative explanation for why all NGs take only 7 inputs from DJ? Why not significantly more or less? If there is significantly more, if there is significantly more, what will happen is that since NG neurons require some threshold for input activity, since it is getting more, if the inputs are more, that threshold is very easy to get crossed. That specific threshold is very easy to get crossed because there are multiple inputs, and since that threshold is easily getting crossed, what is happening is the differentiating power between the orders for the fly will reduce. Therefore, if the inputs, if they are significantly more than 7, it will be bad. It will bad for the fly.
For the first question, when the SP global neurons are artificially blocked, Suhani observed that Joe could not process the orders that were presented to it at high concentrations. Now, it is because SP neurons are involved in the SP neurons actually work at this junction. So what is happening is, when the order is at the higher concentration, so higher concentration would stimulate the DJ layer, but then what is happening is the inhibitory feedback, it would stimulate this, but then the SP neuron belt would not function, and the inhibitory function of the to this sensory and DJ layer junction would not work in the same manner, since they are blocked. So the inhibitory feedback wouldn't be there.
Also, for the fourth C part, what if GRN inhibition is not constant, what will is constant rather than being proportional to input, what will it will reduce the for example, if the input is very large, for example, for a particular case, if the input is very large, since the GRN inhibition is constant, that input amplification will not be reduced, and since the amplification is not reduced, it can affect the sensory parts of the fly, negatively. So since it is proportional, it helps to it helps to adjust the GRN inhibition, and so that it doesn't damage the neural pathway in that sense.
1 minute remaining.
Also, what is the advantage again? What is the advantage the GRN is providing is it is giving proportionally proportional inhibition, basically allowing various distinct odors to get sensed by the fly, since it is proportional to the input rather than being constant.
30 seconds remaining. I request the timekeeper to reset the timer. IIT Delhi, you have 3 minutes to present your answer, and your time starts now.
Yeah, so for question number 1 and 2, basically my claim for how SP works is that you have some smell coming in, and then it is put into some bins of like similar smells by the SP local, and it is fine characterized later by SP global. How this works is that like SP local is then associated with the kinds of VS neurons that are detected in the common odors, which is why it gets very, very active when that particular odor is there. And what SP global does is that, right, when it is getting a lot of input, then it reduces the input behind it. So it reduces the input of, and like this SP local, what it does is it reduces the common input which is there for the similar odors, leaving out the extra like non-common input. And that is my answer to question 1, that this non-common input is then detected by SP global, and which distinguishes the similar smells. So that's and when there are very, very high concentrations, then again SP global, since it gets very, very high value, it reduces all of them, which allows SP local to then fine tune on the lesser smell. Now for question.
So for question 3, we noticed that NG layer takes input from DJ layers, and a combination of firing of DJ neurons or a combination of smells will correspond to a particular odor. By increasing the number of NG layer neurons from 50 to 1000, we are allowing for a wider range of odors to be mapped. The reason 50 is not optimum is that is giving us an upper cap on the number of odors we can detect, 50. For 2000, we have a wider range of combinations of odors, which will help us associate multiple smells with a larger number of odors.
So for 4A and 4B, I drawn like a common thing in machine learning, or also known as the lottery ticket hypothesis, which is that you need a lot of connections or like possibilities for activations, so that you can over time learn the ideal activations for any given thing. So for example, in 4B, why we prefer random wiring is that if you have a lot of random wiring, in this case 2000, then there exists possibilities for detecting the wirings that we want for the smells that are important to us, and also evolve over time. And for 4A, 7 inputs allows there to be a lot of combinations. If there are like for 50 inputs, then you would only have 50 layers and 50 detections, which is not what we want. Like 1, sorry, and so on. No, for 4C.
So for 4C, we notice the GRN prevents spiking in the activity. So we notice neurons can misfire randomly. There's a small chance, but they can. And a misfiring neuron in the NG
30 seconds remaining.
will cause us to associate a particular set of odors, a particular set of smells with an odor. By using the non-spiking activity of GNR, we are preventing those random spikes from the fly learning a wrong set of smells with a particular odor, so to prevent false positives.
The same thing which he said also holds for SP global in reducing spikes.
Time's up. I see you have 2 minutes to present your answer, and your time starts now.
So for the first question, the VS neurons, it says that it gets saturated after a certain time. So our theory is that if you have high concentration of some order, then it may end up saturating your VN neurons if there were no SP global. But SP global manages to attenuate that signal, so the fly is able to sense odors at a higher concentration. It's a similar explanation for question 2. If there are similar smells, similar orders, similar but distinct orders in different concentrations, but both of them are strong enough that it ends up saturating VS neurons, then the SP local neurons end up attenuating that signal and allowing the fly to distinguish between those two orders.
Third part, we can say that why expansion from 50 to 2000, if you have 2000 ng, basically you have a lot of neurons to take input, the output from next layer. Lot of inputs mean that the random errors are going to be reduced, so there is an error correction.
You'll be emphasizing certain portions of the information. So the same information will be expressed in more than
30
one
seconds
neuron.
remaining.
So you'll be repeating that.
And for the if the size were 50, so the random errors would be more because there will be less information.
Okay, so for 4A, we are saying that if you have more than 7 neurons, you are essentially overfitting our model. That is, it will essentially map the data it is taking from the previous layer and just give that further ahead. And for lesser, we are not able to get that diversity of information. And for preferential mapping, we want all sorts of different inputs to be exhibited in our output.
Time's up. IIT Madras, you are up next. You have 2 minutes to present your answer. Your time starts now.
Okay, so for the first question, it is stated that SP Global reduces intensity of input entering, and VS and it is also stated that VS its activity saturates after a point. So what I think is, so like let's assume this is VS, this is sensory input, and this is SP Global. So like SP Global goes and like I said, it inhibits here, but like what it does is it reduces the intensity. So what I think happens is, so like this is like normal input, and the saturation is here. SP Global, the same saturation, SP Global decreases. So like, because like we can figure out the order within the saturation, it's possible, but when like in the question, SP Global is blocked, so we are not able to direct the order, is what I believe.
For question 2, SP Global won't be able to differentiate between two similar yet distinct orders because it has input to all the VS neurons, while SP Local has inputs to a subset of neurons which have similar response. So it's the responsibility of SP Local to differentiate between similar to yet distinct orders.
So for the third question, odors are learned as activity patterns in the ng neurons. So for fine tuning, we will need 2000 instead of 50 ng.
Yeah, so and in the fourth A part, like similar to like what's the role of neural networks in machine learning? Like we have couple of inputs and we have a couple of weights assigned. So if there are less number of parameters, it could lead to more error, as you won't be having like control over all the variables. And if there are more parameters, as the threshold remains the same, all the nodes can become active, and there won't be a sufficient filtering out of those neural networks.
So in 4C, as we know, ng has threshold. So if GRN would not reduce it according to the proportional to the input.
Time's up. I would like to ask the judges if they have any questions for the teams.
Hello. For 4C, basically you have to tell what the functional advantage GRN provides, and what is the if GRN is constant, what will happen. So the first advantage it provides is that it is proportional to the input itself, basically expanding the range of the odor which can be sensed by the fly in the first place. Since if it is the signal is constant, what will happen is if the let's say the odor is very high, the input is very high, what will happen is it can lead the GRN will have a constant inhibition, so it will not completely inhibit the specific odor, and the odor can be very high for the fly to sense at in the first place. So this reduces the range of the odor which the fly can sense if the GRN is constant. But if GRN is proportional, the range is more. The odor range is more.
Okay. And what is your answer to question 2?
For the question 2, we have two types of SP. So that is SP global and SP local. So the DJ layer process, DJ layer can process the concentrated order as well and the less concentrated order as well. So what is happening is when it is stimulated by the SP global, the SP neurons are dampening in the concentrated case. They are dampening the interaction between the sensory and the sensory and the VS neurons. That is what is happening for the concentrated case. And for the SP local neurons in then that case, they share similar order responses patterns. So they work in the when the orders are similar, the response pattern becomes localized. It is not for all of the neurons. So that is how it is working.
Any more questions? No questions? I would request Govind to explain the answer to this question.
Okay, I'll try to keep this short in the interest of time. Nilesh can snatch the mic away from me if I exceed. Okay, so all four of you put together have got the full answer, and I'm very happy a lot of you use the machine learning lingo. But I'll just go to the whole system. So this is the olfaction circuit in the fruit fly Drosophila melanogaster. The first question was about what happens when you remove SP global. You see that you can't learn orders at high concentrations because so you have a singular VS neuron and you have a bunch of sensory neurons projecting into it. If you have a very concentrated order, it will very strongly activate the VS neuron. And since I've told you that the VS neurons input response curve is saturating, if you have too high of input coming from the sensory layer, this will very fast reach here, right? And once it's reached here, there's no distinguishing even if you increase concentration. So if you have a very high order, there's a chance that all your sensory neurons are going to get activated very strongly, and therefore, by extension, your VS neurons are also going to get activated. And your SP global, like many of you mentioned, does the job of separating out the representations. But if all your neurons are in like this regime of input activity, the SP local has no substrate to work on, and it's only really useful if you can be in this range where slight changes in input can change the representation in the VS. So that's what SP global is doing, is doing gain control. When there's not much concentration, it's doing a job of improving the signal to noise ratio. For the second question, it was about how you discriminate orders, but most of you answered. SP global does this volume reduction, and SP local, since it has inhibitory projections from and to VS neurons of similar responses. If you have a signal, two orders A and B that has a representation like this in the layer, it will just turn it into something like this, basically suppress this common part. The third question was, what is the point of the expansion from 50 to 2000 in the NG layer? And that, I think Arvind mentioned, that it creates a larger space to encode orders, but what it does is, you mentioned that there is noise in the system. So, and in the VS layer, there might still be a lot of overlap between two orders, but by projecting that 50-dimensional representation onto a 2000-dimensional one, what you allow is for a much larger ability to separate out those two representations. In machine learning, like things like support vector machines do this sort of stuff. It makes the representation more linearly separable. Fourth question was, why 7 inputs, why not more or less? So if it's less, it's you just won't activate the thing because we mentioned that the input threshold remains constant. If it is more than 7, if it is like 20 or something, a single NG neuron would take inputs from too many VS neurons, and that would by chance activate something, and that would sort of cause a lot of issues, and you would get like overlapping representations because it's not giving you any clear information. 4B, the random wiring, like someone mentioned, it avoids the situation of bias because this whole system is for the fly to learn new orders. So if you have some sort of a preferential mapping where an NG neuron receives input only from a specific kind of VS neurons, then you'll only be able to smell those kind of smells that are preset. But as a fly, you would want to smell any order and attach it to any sort of associative memory. And the last question, what's GRN doing? So the GRN, as you know, is like I think a couple of teams said this. That it's giving proportional inhibitory feedback into the NG cells, and that's basically what's doing is making sure that only the strongest firing neurons in the NG layer are firing. To go to machine learning lingo, this is called winner takes all. And so you represent that order using only the strongest firing neurons, and you ensure that any given representation is only represented by a few neurons. So that obviously allows you to encode more things, and representing an order using only a few neurons in the NG layer means that any unit that comes after only needs to look at these specific neurons to learn any order. About what would happen if GN was constant, the inhibition was constant. If some very weak inputs would just never activate anything in the NG layer, and maybe if your concentration was very strong and you get very strong inputs from the VS neurons, you would activate a lot, and that could again give you overlapping representations, and then whole problem of not learning orders properly. But yeah, I think all of you did a good job with this. So thank you.
Thank you, Govind. That concludes the deep thought round for Biomimamsa. Thank you everyone for attending. We hope you had fun. You have question? We'll continue with Physics Mimamsa Deep Thought at 6 p.m. Thank you everyone.